Welcome back to our final video on Module 9, where we're going to be talking about variations in Mendel's laws. So where we actually see some slight differences in terms of patterns of inheritance whenever we look at alleles and traits that are passed from parent to offspring. And this is going to vary from the patterns of say where you have two alleles or you have episodes of this complete dominance, so where you have a dominant trait and a recessive trait. Now, as we stated, the laws that we previously talked about really work whenever your genes are passed from generation to generation, following those rules of probability that we talked about in the previous two videos. And these laws are really only valid for our sexually reproducing organisms. But now when we look at some other complex variations of patterns of inheritance, we can find that most sexually reproducing organisms don't really account or follow the rules that Mendel has described in the first video of the patterns of inheritance. And a lot of times the patterns of inheritance that we see in the natural world are actually more complex than just the simple dominance versus recessive patterns of inheritance. One example of this is, or this deviation from those simple patterns, is incomplete dominance. And we see incomplete dominance, for example, in flowers, where a flower will have this intermediate trait that is between the dominant and the recessive phenotype. An example of flowers is, say, if you have a red flower that's dominant and a white flower that's recessive, the heterozygous individual will instead be a pink flower, so a combination of the phenotypes for red and white. An example for humans is hypercholesterolemia, and this is a human trait where the heterozygous individual will have a blood cholesterol that's twice the level of a normal individual, where homozygous individuals will have about 5 times the normal amount, so homozygous recessive, and they could actually have heart attacks as early as age 2. And so basically you have an intermediate of the heterozygotes where they don't have as extreme concentrations of cholesterol as the homozygous recessive individuals have, and they don't have the normal amounts of cholesterol like your homozygous dominant individuals have. So they have this intermediate level for the heterozygous individuals. Here's an example of incomplete dominance for flower color. So you have your pure breeding generation, which is shown with the red and white flowers. So you have your red, which is your homozygous dominant, your white, which is homozygous recessive. They can only give off one gamete, which is the dominant and a recessive. Their F1 generation will be heterozygous. So all of that generation will be pink because all of those individuals will be heterozygous. And then if you go through with the F cross, you'll have your 3 to 2 to 1 ratio in terms of phenotypes, because you'll have your homozygous dominant phenotype, your heterozygous phenotype, and then your homozygous recessive phenotype. So your red, your pink, and your white. So the phenotypes for your incomplete dominance matches the genotype ratios. So 1 to 2 to 1 instead of 3 to 1, like we saw with the dominant example. Here we have a figure showing the incomplete dominance of your hypercholesterolemia. And so your homozygous individuals have the ability to make LDL receptors, which help with the cholesterol levels. Your heterozygous individuals have a lower amount of receptors because they only have one dominant allele, whereas if you have 2 dominant alleles, you're making those LDL receptors, so you only have a mild version of the disease. Whereas if you're a homozygous recessive individual, you don't have the ability to make those receptors, so you have really high levels of cholesterol. Okay, so this is an example of incomplete dominance, right? Where those heterozygous individuals have an intermediate version of the dominant and recessive phenotypes. Now, with an example of codominance inheritance, this is where your heterozygous individuals express both phenotypes of the dominant and the recessive as a heterozygous individual. So now with codominance, an example of this codominance is for, say, our blood types in humans. So for humans, we have our ABO blood groups. Okay? These blood groups use 3 alleles to determine the genotypes. Depending on the combination of your alleles, you can end up with 4 phenotypes in terms of blood type. So a human's blood type can be A, it can be B, it can be AB, or it can be O. The letters in the blood types are referring to carbohydrates that are on the blood cells. These carbohydrates are designated as A and B. Okay? And the carbohydrates can be found on the surface of the red blood cells themselves. Okay? And because we have 3 alleles for our alleles that are determining whether or not which carbohydrate is on the blood cell, there's 6 possible genotypes. Okay, and we have them listed in a figure that we'll show on the next slide here. But for our alleles, you have allele A, allele B, and then allele I. Okay, A and B are dominant to I. Okay, so if you have an AI allele combination, an AI genotype, then you're going to have an A blood type because the A is going to be dominant over the I. And so if you also have homozygous A, then you're also going to have an A blood type. Same thing happens with B. If you're homozygous B, you have type B. And then if you are BI, so if you're heterozygous with BI, then you have type B. If you're homozygous I, so if you have that recessive, then you have type O. So the I alleles are what determine the type O blood type. And so you only can have the homozygous I's in order to have an O blood type. So what this means is you can be an A blood type but be a carrier for O, and you could be a B blood type and be a carrier for O. Okay, and people that have an A blood type means that they make the carbohydrate A on the red blood cell. So that carbohydrate is also called an antigen. So they have A antigens on their blood cells. B type blood has the B carbohydrate or B antigens. Okay, whatever antigen that the blood type has or the blood cells have, they make antibodies for the opposite carbohydrate. So if you're an A blood type, you have A antigens, but your body makes B antibodies. If you are a B blood type, you have B antigens, the B carbohydrate, but your body makes A antibodies, which means that if red blood cells with an A carbohydrate enters the blood of a type B person, the antibodies will kind of grab onto those A blood cells and cause those A blood cells to coagulate and clump together in the type B blood. Okay, now the O blood types do not have an A or a B allele, so they don't make any carbohydrates. So they have no carbohydrates on their red blood cells. So they make both anti-A and anti-B antibodies. And so they have both of those antibodies present, which means if you put either type A or type B blood into a person who has type O blood, their antibodies will coagulate and clump together with both of those types. And so that's why only O people who have type O blood can only receive blood from people who have type O. And so that's very important to understand. Now, if we look at type AB blood, they have the genotype AB. So that means that their red blood cells are making both of those carbohydrates. So both of those alleles are being expressed and one is not dominating the other. And so that means that the A is making the A carbohydrate and the B is making the B carbohydrate. So these are said to be co-dominant alleles. So they're both being expressed at the same time. And so the heterozygous AB individuals will have the type AB blood because they have both of those carbohydrates on their red blood cells. So here's a figure showing this relationship. So these are your blood types, A, B, AB, and O. These are the genotypes. You can have homozygous A, or you can have heterozygous AI for type A. This type A blood type is going to have red blood cells with the A carbohydrate or the A antigen on the red blood cell. That means that they're going to make antibodies for B carbohydrates or B antigens. So what that means is if you mix A blood, or I'm sorry, if you mix either one of these blood types, O, A, B, or AB, this is the response you would expect. So O would be okay because it doesn't have any carbohydrates that would bind with the B antibodies. A would be fine because it doesn't have a B carbohydrate to bind with the B antibody. However, if you put any blood type that has a B carbohydrate, which is B and AB, then it will bind with the antibodies and you will get clumping in those transfusions. So you cannot give B blood or AB blood to a person who is type A. Same thing for B. If a person is type B, they cannot receive type A or type AB. And if a person is type O, they cannot receive A, B, or AB because they have both antibodies. However, if a person is type AB, then they have both carbohydrates, and that means that they have no antibodies, so they can receive blood from any type. So AB type people can get blood from any type, but O people can only get blood from O types. Okay, so AB is the universal receiver and O is the universal donor. So O can donate to anybody because they don't have those carbohydrates, whereas AB can take from anybody because they don't have any antigens. So what these relationships mean then is that, again, you have to make sure you have compatible blood groups whenever we have our blood transfusions because of the carbohydrate, the antigen, and the antibody interactions. And so if you have antibodies that will clump with the donor blood cells, then that could be potentially fatal for the recipient. And so you have to make sure that you get those blood types right. And so with that, we have a question here. So if Maria has the O blood and her sister is AB, what are the genotypes of the girl's parents? Okay, so take a few minutes to try to figure this out, and then pause the video and we'll go over that answer. Okay, so if we know that one of the sisters is AB, okay, and one of these sisters is O, then that means that the sister who is AB has to have the alleles A and B, and the sister who is O has to have the alleles I and I, because that's the only way you can have an O blood type, and that's the only way you can have an AB blood type. So what this means then is that their parents have to be, if we work backwards, one parent has to be carrying A, one parent has to carry a B. That's where the AB will come from. And then each parent has to be a carrier for the O allele. So they are heterozygous for AI and BI, because you'll get A from one parent, B from another parent, and then an I from one parent and an I from another parent to give you your O blood type. And so that would be the genotypes for the parent. Okay. Now the next thing we're going to talk about is pleiotropy or pleiotropy, pleiotropy and sickle cell disease. So pleiotropy is whenever you have one gene that influences several different characters. So for example, sickle cell disease exhibits pleiotropy. And pleiotropy is where you have, sickle cell disease is where you have abnormal hemoglobin proteins. And this causes an abnormal shape in your red blood cells. So for example, a normal hemoglobin protein has a concave disc-shaped hemoglobin, whereas a sickle cell protein has a sickle cell shape, and it has these little jagged edges. And so that shape causes the blood cells not to flow smoothly through the blood vessels, and they can actually accumulate and clog in those blood vessels. And it also alters how efficient those red blood cells will carry oxygen around the body. So here we have an example of an individual who is homozygous for a sickle cell allele. So they're homozygous recessive. So they have sickle cell hemoglobin, and then that hemoglobin has this particular shape. And then the sickle cells can have a bunch of different symptoms where you have weakness, pain, organ damage because of lack of oxygen, and also paralysis in certain situations. Okay. And so what the pleiotropy is then is where you have one gene affecting several different characters. Okay, so it's affecting the abnormal hemoglobin and the ability of the hemoglobin to carry oxygen efficiently around the body. Another type of inheritance that we can see is a polygenetic inheritance. And this is where you get additive effects of multiple genes interacting with one another in order to affect the phenotype that we see in an organism. And this is the opposite of pleiotropy, in where pleiotropy has one gene affecting several characters, but polygenetic is multiple genes affecting one trait, okay, or one character. And an example of polygenetic is height. So height in people are controlled or is controlled by several different genes, and you inherit those genes separately, okay? Other examples are things like diabetes, heart disease, and cancer, which will follow a polygenetic inheritance pattern. And one thing we see with polygenetic inheritance is that a new property can come up as you have interactions of many different genes. So if you have genes that weren't interacting before and they interact later or now, they could bring about new characters or new traits in the population. So here we look at an example of polygenetic inheritance where we have, say, 3 alleles affecting, or 3 genes affecting height. You have recessive, and then you have dominant for each of those 3 genes. So you have dominant A, a recessive A, a dominant B, a recessive B, a dominant C, and a recessive C. So if you have recessive for all 3 genes, then you're the shortest you can be in that population. If all three of your genes are dominant, so homozygous dominant, then you're going to be the tallest in that population. So you'll be on the extremes. Now, if you have various combinations of these, then your height is going to vary based off of this distribution. And so, for example, if you only have one recessive allele, right, which is shown here. So if the circle is colored black, then those alleles are all dominant. If the circle is colored white, then that allele is recessive. So for example, here you would have homozygous A, so dominant A, recessive A, heterozygous A, dominant A, recessive A. Then you would have homozygous dominant B, homozygous dominant C. So only one recessive allele. And so you would fall in the next tallest category. And then you could have any one of those combinations in heterozygous C and heterozygous B. And then below that, you would have two recessive alleles, then three, then four, then five, and then six would be the shortest, where you would have all of your alleles being recessive. And so that gives you kind of your distribution of height in the population. And so again, it's the culmination, the additive effect of all of those genes that determine how tall or short an organism is. Now, adding to the complexity of these inheritance patterns is the fact that not only is our phenotypes determined by genetics, but our phenotypes are also determined by environment. So we have this genetic and environment interaction that ultimately determines what our final phenotype looks like. Okay? So we have many phenotypic characters that result from not only a combination of heredity, but how the environment acts on that organism. And so you can have human characters that could be more influenced by genes or more influenced by the environment, which is what we mean by nature or nurture. And depending on the type of environment you're in, can determine kind of the phenotype that an organism has. So for example, if you have two identical twins that have the same genotypes and they grow up under different environments, their resulting phenotypes could be different. And so for example, if you have a plant that has seeds and all of those seeds are genetically identical, and you plant one seed in a hot environment and you plant one seed in a cold environment, say the plant in the hot environment may grow to be a tall individual. The plant in the cold environment may grow to be a very short individual. Even though they have the same genes, the height of the plant is determined by the environment in this example. So not only is it the genetic influences that affects the phenotype, but it's also the environment that those organisms grow in. So even though you have identical twins, the environment that they're exposed to can actually lead to phenotypic differences between those organisms. Another factor that's really important is called epigenetics. So epigenetics is basically the expression of the DNA sequence or the gene. Okay, so for example, we know that you have parts of chromosomes that are tied to a specific DNA sequence and a specific gene, and that gene in turn codes the development of a protein. Okay. Modifications in epigenetics basically say that the gene activity can differ and lead to different phenotypes. So for example, if you have a gene that is expressed early on in the embryonic development stage, the phenotype can end up being different than if the gene is expressed later in development or through the whole period of the development. And so when that gene is expressed and for how long that gene is expressed can ultimately determine the phenotype. And by gene expression, we're basically saying when that gene is turned on. So when it is actively making proteins. And also you can have more expression versus less expression. So that gene can be more active or it can be less active, which will ultimately determine what that organism looks like. Now, we also have the idea of the chromosome theory of inheritance. So this theory states that genes located at specific positions on a chromosome and the behavior of the chromosome during meiosis and fertilization determines our inheritance patterns. And we've talked a lot about this. We've never just quantified it as the chromosome theory of inheritance, and that's kind of what we're doing now. We're basically saying that this theory says how those chromosomes separate in meiosis, how fertilization happens, is responsible for your inheritance patterns. And then remember, it's the chromosomes that segregate and assort independently during meiosis. And so that accounts for the Mendel's laws that we talked about in the first video. And the next figure shows the culmination of these different laws and how you end up with your different patterns of inheritance. So for example, if you have a dihybrid cross with round yellow seeds and wrinkled green seeds, we mate these pure breeding organisms together. You get your heterozygous organisms, which are all round yellow seeds. You cross individuals from these heterozygous F1 generation. This is how your gametes can sort, right? You can have this type of independent assortment or this type of independent assortment, right? Where they switch, along the metaphase plate. And then you have the law of segregation saying that your alleles are going to segregate independently of one another, meaning that just because you get a capital R doesn't mean you get a lowercase r, and vice versa. Or just because you get a capital R doesn't mean you get a capital Y, and vice versa. And so what you end up with in the end are these four possible gametes where you have a capital R, lowercase y, lowercase r, capital Y, lowercase for both, and then capital for both. And so you get these different gametes, and then random fertilization. So it could be 25%, capital R, capital Y, fertilizing with capital R, lowercase y. And that ultimately determines your 9:3 to 3:1 ratio, phenotypic ratio that we see at the end of our dihybrid cross here. Okay, so some other factors and instances of inheritance patterns that we're going to talk about is linked genes and sex chromosomes. So inheritance via sex chromosomes. Linked genes is this interesting relationship where you'll have genes that are located near each other on the same chromosome. So basically right next to one another. And this means that when crossing over happens, that it doesn't really separate those genes or those alleles. And so they tend to travel together through meiosis and fertilization. So linked genes are often inherited together and don't follow that law of independent assortment. So for example, let's say we have a chromosome, right? And on that chromosome, we have two genes, right? One gene here and one gene here. Okay, now these genes are really close together. Okay, so that means when crossing over happens, so like if we cross over here and we attach it onto a different chromosome, these two genes, these two alleles are probably going to go with one another. So let's say it's capital R, capital Y. So anytime you have linked chromosome or linked genes, not chromosomes, anytime you have linked genes, that means that they're going to go with each other. So in this case, if you get a capital R, you're going to get a capital Y because they're located next to each other on a chromosome. However, the farther apart those genes are on a chromosome, okay, now let's say these genes are located like this, right, where you have the Y now down at the bottom and the R now at the top. Okay, so even though they're on the same chromosome and the chromosomes, whatever alleles is on the chromosome goes together, but you do have this chance to where crossing over can happen and actually separate the capital R and the capital Y. So now when recombination happens, you could say, for example, get a lowercase r when crossing over happens, right? And so you'll have a lowercase r and a capital Y. And so now you get a variation here, and these genes aren't necessarily linked, even though they're on the same chromosome. And so this can happen to where you do have linked genes and they don't independently assort from one another. Your independent assortment really happens when the genes are located on different chromosomes. And so what we mean by that is if you have a chromosome that has, say, your homologous chromosomes carry the R's, so let's say it's like this, and then you have another chromosome, right, your homologous chromosomes, that specifically carry your Y alleles. Okay? Now these will definitely always independently assort from one another because they're not on the same chromosome. Okay, and so now what we're going to look at next, aside from these linked genes, are sex chromosomes. Okay, and so sex chromosomes, remember, are the last pair of chromosomes that we have in our 23 pairs of chromosomes. And so with those other 22 pairs of chromosomes, those are your autosomal chromosomes. So those are the chromosomes that are found in all of your regular somatic cells, so your autosomes. Your sex chromosomes are found on your gametes, right? Your sex cells. And so what we have with our sex cells, our sex chromosomes, is that individuals will have either an X chromosome and a Y chromosome if they're a male, or they will have two X chromosomes if they're a female. Okay, and so that means you have 22 pairs of autosomes, which are your 44 chromosomes that are autosomes, and you have one pair of sex chromosomes, which is the other two chromosomes, which total to 46, so 44 plus 2. And so what we have now is this relationship here where if you're a male, one of your gametes will have 22 chromosomes plus an X, and one of your gametes will have 22 chromosomes and plus a Y, because you'll give either an X or a Y in your gametes. Whereas females will always give an X because they're homozygous X. So they'll have 22 chromosomes and an X regardless. Okay. So based off of the combination of which sperm fertilizes the egg, you'll either get 44 chromosomes with two X chromosomes, or 44 chromosomes with an X and a Y, which will result in male offspring or female offspring. Okay. Now with sex-linked genes, so genes that are only found on these sex chromosomes, so only on the X chromosomes or the Y chromosomes in some cases, but most of the time the ones we'll talk about are only found on the X chromosomes. So these sex-linked genes are found on the X chromosome. Again, so most of them are on the X, very few are on the Y. And so a number of conditions like color blindness comes from these sex-linked recessive alleles. Another condition is hemophilia, so you're hemophiliac, so like where your blood doesn't clot appropriately, and then also muscular dystrophy. And so basically you can look at the sex chromosomes and determine what alleles you have to determine if you're a carrier or if you have one of these types of disorders. So for example, red-green color blindness is a very common sex-linked disorder. That's where you have this malfunctioning, so dysfunctioning of light-sensitive cells in the eyes. Most of the time your males are affected, but your heterozygous females can have some defects too in terms of color blindness. So this figure here basically is a colorblind test that your doctors will use. And mainly it's looking to see if you're red-green colorblind. So if you can't pick up the green circles in this chart or this picture, so you should be able to see a group of green circles here that make out the number 7. And so this is again a test that doctors will use to tell if you are red-green colorblind. And so what that means then, or what we can look at, is how exactly is these color or sex-linked genes passed on from generation to the next, and how does that color blindness actually show up if these alleles or genes are sex-linked. Okay, so if we say that these alleles, or if this is a sex-linked trait that is found only on this X chromosome, then the X chromosome will only carry the alleles for the gene, or for the trait. So females will always have 2 copies of these alleles because they have 2 X chromosomes. Males will only have 1 copy of the allele. So what that means is that males will either have the disorder or they won't. So if this is a recessive disorder, so if blindness is a recessive disorder, then a male will have only 1 X chromosome. And if the allele on the X chromosome is recessive, like we see here, the lowercase n, then the male is colorblind. If the allele is dominant, like we see here, then the male is not colorblind. So they either have it or they don't. So they either have the allele or they don't have the allele. With females, it's different. It's like the dominance, the complete dominant and recessive traits. So if the female is homozygous dominant, then she does not have the disorder, and she is not a carrier because she cannot pass on that recessive allele. However, if she is heterozygous, then she is a carrier for that allele. Okay, so now we've kind of discussed how the traits are going to be expressed, right? So your males either have it or they don't, so recessive or dominant. Females can not have it and not be a carrier if they're homozygous dominant. They can be a carrier if they're heterozygous, so they have both dominant and recessive alleles. And then they will show the disease if they're homozygous recessive, so if they have both of the recessive alleles. Okay, so that is the situation for both the females and the males. Now what we can do is we can do crosses with our Punnett squares to determine the probability of the offspring having the disease. And you can also look at it in a couple of different ways. You can say, if my offspring is a male, what is the probability they have disease? If my offspring is a female, what is the probability they have the disease or not? Or you can say, in general, what is the probability that I have a child that has a disease or not? Okay, so if we look at this first cross here, you have a female who does not have the disease, and you have a male who does have color blindness. So the male is always going to pass an X that has the color blindness allele. The female is always going to pass on an X that does not have the color blindness allele, so the dominant allele. So in this case, you'll have all of your females being carriers, because all of the females will get an X from the male and an X from the female. So the X's will always be dominant, and then one X will always be recessive. So all of the female offspring will be carriers. The males will not be colorblind because they will get the Y from the male and an X from the female. So they will never have it. So 50% of the offspring will be carriers, 100% of the females will be carriers, and 100% of the males will not be colorblind. Okay. Now, if we have a cross where the female is a carrier and the male is normal, then we can end up with a little different situation where 3 out of 4 of the offspring will be normal vision. 25% of my offspring will be a carrier, and so 50% of the females will be carriers, because 1 out of 2 females will be normal, no carrier, or a carrier. But then 50% of the males will have color blindness. So 50% will be normal, 50% will be colorblind. But in total, 25% of the offspring will be colorblind. Now, if you have a carrier female with a colorblind male, then you'll have 50% of your offspring being colorblind, 50% being normal, but 50% of your females will be carriers, 50% of the females will be colorblind, 50% of the males will be normal, 50% of the males will be colorblind. Okay, and then 25% of your offspring will be carriers. Okay, so there's a couple of different ways we can go about describing that pattern, but the key thing is remembering that your males will either have the trait or not, because they only have one X. The females can be normal and not a carrier. The females can be a carrier and normal, and then the females can express the trait if they are homozygous recessive. And another example of sex-linked genes is hemophilia, and it's a sex-linked recessive trait, just like color blindness. And it's got a long documented history because, like your royal bloodlines, they wanted to keep the bloodlines pure, so they tended to keep marriages within the royal families. And so that means that there was some inbreeding that would go on, and that the hemophiliac trait was kept in the family line because of that, because you wanted to strengthen those international alliances and the royal alliances. So you would marry within royal families, and so that led to some instances of inbreeding and kept those traits going through the families. And so what you would end up with is kind of strong examples of where you would have expression of that disease within the family because of the way that those families mated between those lines. And so, for example, a royal family in Russia had a strong incidence of females being carriers, and then eventually you had Alexis expressing the hemophilia, which is an example of 3 generations here. Okay, and so that's it for our discussion on genetics. We have finished our 3rd and final video for the module. And so make sure you go through and complete everything that you have to do with the module, including the lab that's on genetics and the lab quiz. And as always, let me know if you have any questions, and we will see you in the next module.